Today we're going to learn about karyotypes, what they are, how they're made, and how they can be helpful. Okay, so maybe you've already kind of figured it out by looking at this. What are we looking at a picture of? Chromosomes. Okay, so a karyotype is a picture of human chromosomes that we can analyze. Okay, and this picture is taken during mitosis because that is when the chromosomes have condensed from their chromatin form into actual chromosomes, so we can see them with like a microscope. Okay. So the scientists will take the picture, but they're going to be all jumbled up. They're going to be all over the place. And digitally, they will kind of start arranging them. How do they arrange them? Well, they put them in order from chromosome 1 to 22, and then the sex chromosomes. And part of what helps them do this is they stain them. They apply a dye, and that dye attaches itself to different segments of DNA. And so chromosome 1, for example, will have a certain dye pattern, of a certain banding pattern. And we have two copies, of course, one from mom, one from dad. So the scientists can look at the length and see, hey, these two are similar size, and also look at the banding pattern and say, oh, yep, these match up. These are chromosome 1. Okay, these match up. They're chromosome 2, and so on and so forth. Another thing that you might notice here is this little dotted line shows where the centromere is. So another way that scientists can help organize the chromosomes is by looking at the centromere location. And so the centromere can be at different points. We typically think of it kind of as like this or this, right? Somewhere close to the middle, but it can actually be in several different places. The centromere, if it's legitimately right in the smack middle, these little gray dots of the centromere, that is called metacentric. If it's slightly off center, that's called submetacentric. If it's pretty close to the tip, but not quite there, it's called acrocentric, excuse me. And if it is legitimately at the tip, right at the end, it's called telocentric. Okay, and so if you look back, notice here, right, this would probably be metacentric. This maybe be sub. Here's acro. Here's maybe right at the tip, right? So we've got some different positions of our centromere, and that can be helpful for organizing the chromosomes too. Okay, so how do they get, what kind of cells do these come from? How do they get these chromosomes? Well, from an adult, you can use any old cell really that has a nucleus, right? So blood cells are a really easy one. Skin cells, if you do a biopsy of a piece of tissue, whether healthy tissue or a tumor, that's where you can get the cells where they can take the pictures of. We can also do karyotypes on babies that are in utero that haven't been born yet. So we can do some sampling there. And this process obviously might be a little bit tricky. There's two different ways you can do this. One is called an amniocentesis, or referred to as an amnio maybe. And so what happens is the doctor takes an ultrasound probe and places it on the mother's belly. And so there's a screen showing the image of where the baby is. Okay. And then a doctor takes a big, gigantic needle and sticks it through the woman's skin, goes all the way through her uterus, through the placenta, inside to where the amniotic fluid is. That's the fluid like cushions the baby. And they take a small sample of this fluid, and they use this ultrasound so they don't accidentally poke the baby, because that would obviously be bad. And then from that amniotic fluid, there are baby's cells in there, and so then they can look that way. This can be done a little bit later in pregnancy. I don't know, starting around maybe 4 months, 16-ish weeks is when there's enough amniotic fluid that they can do this and be safe. If you are worried about issues before then, you can do what's called CVS, or chorionic villus sampling. Okay, and it's kind of a similar process, but they can't use a long needle and get the amniotic fluid because there's not enough of it yet. There's not enough room in between the little baby, and like there's not enough amniotic fluid to do that. But basically, instead of going in through the belly, they go in through the vagina, and they use a catheter, which is like a little tube, and they go up into the uterus and they collect a little sample of the placenta of the villi. Okay, the like edge of the placenta. And they, again, use an ultrasound probe so they don't hit the baby, but they collect some of the placental cells and bring that back out. And again, that will have baby cells in them. And so then they can do the sampling there. This can be done almost like a month earlier in pregnancy. So, you know, if you need results earlier, you might do this. If things are happening later in the pregnancy, then, you know, they could do this. Both of these have some risks to the baby, obviously, but, you know, and it's up to each family to decide if the risks are worth it to proceed forward with the procedure. Anyway, so once we get those pictures of the chromosomes, we can figure out how many chromosomes there are, right? We can figure out if there are the 46 we're expecting. We can figure out the gender of the baby, which is kind of cool. And actually, these days, if you take a sample of the mom's blood, there's a little bit of the baby's mixed in there. And so you can actually tell the gender of the baby from taking a mom's blood sample, which is kind of cool too. I don't know if they can do a full karyotype, but I know they can at least determine gender now, which is pretty sweet. And so you can take a look at this and you can see kind of what's going on with the individual. We can look and see if there's the right number of chromosomes and if they look like they're the right shape. So for example, for this one, we see that there's 3 copies of number 21. And this is called trisomy 21, and this is also known as Down syndrome. Okay, so we could look at this. Now, if the person's already an adult, we would know that they would have Down syndrome, but if they're a baby in utero, we would know before they were born that they had Down syndrome, for example. Okay? We can also, with the karyotype, see if there are any mutations of the chromosome. So there are different things that can happen to the structure. Like there's a deletion where like an entire segment right here, D, just boop, disappears. Or a duplication. You might have a part of a chromosome that's repeated a bunch of times. Or an inversion where a part of a chromosome is flipped around. You can also have a translocation where part of a chromosome, like this MNO, where the AB was, MNO joins in and it goes MNO, C, D, E, F, G, H, right? So you can get segments moving from one chromosome to another. And if this happens, this can lead to genetic problems, okay? So one example, another cause of Down syndrome, we learned that it can be from 3 copies of chromosome 21, but it can also be from a translocation between 14 and 21. So basically the end of chromosome 14 and the end of chromosome 21 break off and join together to form their own new chromosome, and that can be a cause of Down syndrome, and that could be identified from a karyotype. Let's take a look at a couple practice to see if you can read these. So here is human karyotype, and what would be the gender of this individual? See down here with X or Y, that would be a male. How about down here? This karyotype is abnormal. What is the thing that's different about it? It only has, notice here, there's 1 X and no Y. It should either be XY or 2 X's, okay? And as it turns out, this is called Turner syndrome. And so this ends up having the individual be short, delayed puberty, infertility, things like that. Sometimes learning disabilities. Here's another one. What's going on with this one? Can anyone find it? This is a female, but look at this, 3 copies of chromosome number 18. This is called Edwards syndrome. This is really, really devastating. Low birth weight, small head, defects in the organs, and a lot of these babies don't even make it till birth, or they die really, really young. Okay, so now hopefully you've understood what karyotypes are, how they're obtained, and some different info we can find from them.